So I'm going to finish talking about the only thing I didn't talk about yesterday on energy. It'll just take me a couple of minutes to do that. And then I'm going to talk about structure of carbohydrates. So that's what's on tap for today. I pretty much went through everything that was critical yesterday with respect to energy. I didn't talk about that, but I sort of implied that nutrients we break down to get energy to make ATP, and the ATP we use to make the things that we need, proteins, fats, carbohydrates, and so forth. One of the things I didn't point out to you is the fact that the electron carriers that I talked about yesterday, that is NAD and FAD and NADP, the electron carriers have to be recycled. That is, when you take NAD and you make NADH, that NADH has to dump its electrons off to something to become NAD again. Okay? And we'll see that happens in the electron transport system. That's actually what's shown down here, where the reduced electron carriers, and you can tell they're reduced by the fact that they have an H on them, the reduced electron carriers dump their electrons off. They also dump their protons off and regenerate NAD and FAD, which then go back and be used in oxidative processes inside of the cell. Okay? So that's a really important thing to remember. We have a limited amount of electron carriers. And for those of you who are interested in things like fermentation, that's a very important consideration, because the reason that cells ferment is because they've run short on oxidized electron carriers. Okay? They've run short on oxidized electron carriers. We'll see that as we get talking about metabolism. But this electron transport process is very important in that respect. Okay, so that's the last of what I wanted to say about energy. I want to turn our attention now to talking about the structure of carbohydrates. So I'm going to go through this fairly quickly, partly because you've seen this before in organic chemistry, and partly because it's mostly just nomenclature. There's a little bit of structure associated with it, but most of it is nomenclature. So, carbohydrate. What does the word mean? Well, the word carbohydrate means carbon, carbo part, and hydrate means water. So carbohydrates are molecules that have a carbon with water, basically, is what it means. And when we see their structures and when we see their formulas, we realize why that's the case. Okay? The shortest carbohydrates that we talk about here are the 3-carbon carbohydrates. Okay? Glyceraldehyde and dihydroxyacetone are both carbohydrates. And if we were to write the formula for them, we would see the formula would be C3H6O3. And that could be written as C with parentheses water, and then a parentheses around that, and 3. So it's really a hydrate, that is, it's water that's been added to carbon. That's literally what a carbohydrate is. Okay? Well, carbohydrates can have many carbons. We can have simple carbohydrates, we can have complex carbohydrates, and so forth. Okay? Another term that we use interchangeably with this is saccharide. Okay? Saccharide. So saccharide from, I think, the Greek, I'm not sure of that, means sweet taste. Okay? Sweet taste. So saccharides are things that taste sweet. We think of sugars, of course, as saccharides. And these come in also, again, simple forms, monosaccharides, or complex forms, polysaccharides. All right? So those are some terms that we hear a lot about. All right. Well, when we talk about the simple saccharides or the simple carbohydrates, as I said, we typically start with the 3-carbon ones. And 3-carbon, oh, by the way, there's another thing I should tell you about nomenclature. And the nomenclature of this is anytime you see the letters OSE at the end of a name, it refers to a carbohydrate. OSE. So ASE at the end meant it was an enzyme. OSE means it's a carbohydrate. So we see, in fact, these guys are the simple ones. We don't even, their names predate that designation. But we do refer to them by their general name of triose. All right? So a triose is something that is a sugar or is a carbohydrate that has 3 carbons, and these guys have 3 carbons. So these guys are both trioses. Something that has 4 carbons we refer to as a tetrose. Something that has 5 carbons we call a pentose. Something that has 6 carbons we call a hexose. Okay? So those are general categories. As we will see, there are many hexoses. There are many pentoses. Okay? But there are only 2 of the trioses. Actually, that's not completely true. There are 3 if we consider stereoisomers. Okay. Stereoisomers come into play. We've talked about stereoisomers before, reminding us that any carbon that's bound to 4 different things have those 4 things arranged in 3-dimensional space in 2 different ways. And here's where biochemists being lazy really makes things work, because we have very simple naming conventions for how these things are named. You see this is called D-glyceraldehyde, this is called L-glyceraldehyde, and you see that it relates to the position of this hydroxyl being on the right side or the hydroxyl being on the left side, as you see it here. Okay? Now, we'll see that more longer or bigger carbohydrates can have more than 1 asymmetric carbon, but you'll see that the nomenclature for those is actually quite simple also. All right. Well, here, for example, is glucose. All right, and we see that glucose, we've added another term here, is an aldose. Okay, so an aldose, again, ose referring to a carbohydrate. An aldose is a carbohydrate that has an aldehyde at its end. All right, an aldose is a carbohydrate that has an aldehyde at its end. So glucose is an aldose. Glucose is also a hexose because it has 6 carbons. And we see it's in the D configuration because the next to the bottom carbon has the hydroxyl on the right side. Okay? D-glucose. This is D-fructose. Fructose is what we call a ketose. And a ketose is something that instead of having an aldehyde at its end, it has a ketone, most typically at carbon number 2. Okay, so that is a ketose, and that should be an O. That's not an OH. That should be a double bond O. I just noticed that. Sorry about that. That's not an OH. Double bond O. Sorry. Okay, so that is fructose. It's also in the D configuration because, again, that next to the last carbon at the bottom has the hydroxyl on the right side. Any sugar that has that next to the last carbon on the right side is in the D configuration. Now, next I'm going to show you something that's a little confusing. So I want you to pay attention to it, because even biochemists who've been practicing biochemistry for a long time mix this up sometimes. Okay? The L configuration of glucose does not simply take the hydroxyl and put it on the left. The L configuration of glucose is the mirror image of D-glucose. That means all of the hydroxyls have to be flipped. This guy that was on the right goes to the left. This guy that was on the left goes to the right, right to left, right to left. Okay? So L-glucose is the mirror image of D-glucose and vice versa. Okay? We give the term enantiomer to describe a mirror image. So if I said, what is the enantiomer of D-glucose, I would hope you would tell me it is L-glucose. If I said, what is the enantiomer of D-fructose, you would say it is L-fructose. Make sense? So enantiomers are mirror images. Okay. I told you that we get more carbons, we can have more asymmetric carbons. In fact, every time we add an additional carbon onto these, we create yet another stereoisomer. So whereas with the 3 carbon guys, we only had 2 stereoisomers that was of glyceraldehyde, that was D-glyceraldehyde and L-glyceraldehyde, because dihydroxyacetone was a ketose, not an aldose. The adding of additional carbon here means now that instead of having 2, we have 4. So we have D-erythrose and its mirror image, which is L-erythrose. And no, you don't need to know these names or these structures. I'm just showing you examples. And here is D-triose, and L-triose is the mirror image. Okay? Now, there are some sugars I'm going to expect you to memorize the structure of. Okay? I didn't ask you to memorize structures of amino acids, But there are some sugars I'm going to ask you to memorize the structure of. And the reason I do this is not to be mean or give you something more to memorize, but because when you're taking further classes along, I can guarantee you your professors are going to expect you to know them. So the second time you memorize them will be much easier for you. So what I'm trying to tell you is I'm doing a favor for you. You're supposed to say, whoopee. Thank you. Thank you. Okay. I like it when people say whoopee. All right. So anyway, I will tell you what they are. I think you should know the structures of glucose, fructose, and ribose. Now, as we will see, there are some considerations for those structures, whether they're in the straight chain form like we see on these guys, or if they're in ring form, which I'll show you in a bit. You need to know both of those, straight chain and ring. Okay. Here shows the progression of the D aldoses as we go. This is just the D now, not showing the L's, just showing you the D's of a 3-carbon, a 4-carbon, a 5-carbon, a 6-carbon. To get the total number, we'd have to double these, because that means we'd have 2 of the 3 carbons, 4 of the 4 carbons, 8 of the 5 carbons, and 16 of the 6 carbons. Okay? Now, you can see their structures. Again, you don't need to memorize all the structures. There's ribose right there. Ribose is very easy. Look at that. It's an aldose. It's an aldose that obviously has 5 carbons, meaning it's an pentose. And all the hydroxyls are on the same side. Very simple. Okay? Here's glucose. Okay? Glucose is an pentose. You can memorize the structures a variety of ways. I remember it as right, left, right, right, where the hydroxyls are. Right, left, right, right. Okay. This is hexose, right? It's got 6 carbons. Okay. And this is showing the aldoses. So fructose isn't appearing on here because fructose is a ketose. Okay. There's some other common sugars that are on here you don't need to know. One is galactose, shown over here. Okay, galactose right there. Xylose is not an uncommon sugar. It's also there. And arabinose right there also shown. Okay. Now, one of the things that I find that most books don't have that I don't like is they don't have good structures for fructose and sucrose. So I've drawn fructose for you in a ring structure. I've got it up in a ring structure for you right here. Okay. I thought I had linked that to one of my own drawings, but I'll link it to a drawing later tonight so you'll have that. That's what fructose looks like. Okay. That's actually a carbon right there. So fructose is a 6-carbon. I'm getting ahead of myself. Let me come back to that. It's a 6-carbon molecule, but it's showing you a ring before I've shown you how to do rings. So I've got to go back and do rings. Okay, let's do rings. How do we make rings? Well, it turns out that for molecules that are for carbohydrates that have 5 or 6 carbons, the chain is long enough that they can actually loop back on themselves quite readily. And they do that. And when they do that, they form a ring structure. So you can actually see here's the linear form of glucose, and here it is forming a ring. Okay? So we see that this carbon number 1 is coming back very close to, this carbon number 1 is coming very close to the hydroxyl of carbon number 5. And that's what's happening here. This forms a ring structure. And the ring structure that it forms is shown here. We see that in the process of forming the ring, a new hydroxyl group appears. And that happens because the hydrogen here goes on to that oxygen and makes it a hydroxyl. That creates a carbon that has 4 things on it. This carbon up here only had 3 things on it. That carbon number 1 now has 4 things on it. That's a new asymmetric carbon. So this asymmetric carbon was created as a result of forming a ring. It was created as a result of forming the ring. This asymmetric carbon is given a name. It's a brand new one. It's called an anomeric carbon, A-N-O-M-E-R-I-C. O-M-E-R-I-C. Anomeric carbon, okay, is the newly formed asymmetric carbon made by making the ring structure. Well, because it's asymmetric, it means it can have two different configurations. One configuration is called alpha, and the other configuration is called beta. The alpha configuration has the hydroxyl in the down position, as you can see here. The beta has the hydroxyl in the up position, as you can see on the bottom figure. We call this ring structure that I've drawn here the Haworth projection. The Haworth projection. Now, some people call this type of a structure a Fisher projection. I tend to call this on the left a Fisher projection. Okay? That is, this indicates there's a ring, but Fisher actually doesn't indicate a ring. So either one, I'll take on an exam if you call this a Fisher or you call this a Fisher. But if you draw it in this configuration, then you have to put it into, you have to actually draw the ring around there. Okay. Now, the last nomenclature thing I'm going to give you on here is the nature of the ring itself. This, as you can see, this ring has 6 members, and here's the 6 members on the glucose ring. That oxygen plus carbon number 1, 2, 3, 4, 5, and carbon number 6 is not part of the ring. You see, it sticks up in this configuration. Okay? It sticks up. All right. 6-membered rings are called pyranoses. So glucose in a 6-membered ring is called a pyranose, and that's what pyrin looks like right there. So it resembles pyrin, and so it's called a pyranose. Okay. Now, that's repeating the same thing I just told you, so I'm not going to show you that again. And here are some Haworth projections, okay? So, Haworth projections, those are pyranoses up here, all right? Here are furanose projections up here. It's possible for a hexose, like fructose, to appear in a furanose form. Furanose means 5, F for 5, 1, 2, 3, 4, 5. And when we have that for fructose, okay, that means carbon number 1 is sticking above the ring, and carbon number 6 is also sticking above the ring. Okay? So I'll draw a figure for you tonight and give you the fructose in that configuration that you can see tonight. Okay? All right. Now, for fructose, the anomeric carbon is not carbon number 1, it's carbon number 2. It's the one that had the double bonded oxygen. The anomeric carbon will always be the carbon that had the double bonded oxygen. In the case of an aldose, that's carbon number 1. In the case of a ketose, that's carbon number 2. So for this furanose that's here, all right, and I just don't like that figure. Again, I'll draw that for you tonight and show you that. Okay? Okay. But for a furanose, it's carbon number 2. And carbon number 2 is actually right there. I don't like that number being right there, because carbon number 1 is up, carbon number 2, 3, 4, 5, and 6. Okay. All right. Those are Haworth projections. Now, sugars are reactive. Sugars are reactive. One of the things that you learned in organic chemistry, I hope, is that aldehydes are fairly reactive. They're fairly reactive. They oxidize fairly readily. And glucose, being an aldehyde, an aldose, is also fairly reactive. Okay? It's fairly reactive. It's fairly reactive because this ring structure that we see right here, you'll notice in the ring structure, we don't have any aldehyde at all. But this ring structure can open up, and when it does, it forms an aldehyde. That aldehyde can be oxidized. And when something gets oxidized, I told you yesterday that something else gets reduced. Consequently, we refer to glucose as a reducing sugar because it can form an aldehyde very readily and it can get reduced. In fact, anything that can form an aldehyde readily will get oxidized and cause something else to be reduced. That means that anything that can form an aldehyde is going to be a reducing sugar. Any sugar that can form an aldehyde, any aldose will be a reducing sugar. The words aren't coming to me today. Okay? Any aldose can be a reducing sugar. Ketoses, on the other hand, are not so readily oxidized and are not very much of reducing sugars. Fructose is not a very strong reducing sugar because ketones are not oxidized nearly as readily as aldehydes are. Okay. If we take a sugar and we reduce it, that is, if we take, in this case, its ketone and we convert it into a hydroxyl, if we do that, we create something called a sugar alcohol. A sugar alcohol. They're also called alditols. Okay? Alditols. These alditols have some properties kind of like sugar. In fact, artificial sweeteners, sometimes like sorbitol here, taste sweet because they have a structure very similar to sugar itself. Okay? Structure very similar to sugar itself. Xylitol is also used as an artificial sweetener, and it looks like a sugar as far as our taste buds are concerned. Okay. Sugars can be phosphorylated. We'll see how that happens in the lecture on glycolysis, meaning that they can gain a phosphate. And when they gain a phosphate, they actually gain some fairly different properties. All right? So here is beta-D-glucose. Beta referring to the fact that hydroxyl is up. It's in the D configuration. In fact, in this class, we will almost never work with a sugar in the L configuration. Almost never. Okay? It's in the D configuration. And no, I'm not going to ask you how to draw the Hayworth of an L sugar. I might ask you to draw the Fisher of an L sugar, but not a Hayworth. Okay? Here it is in the D configuration. It's in the beta configuration here. A phosphate is being put onto it. Here's the phosphate coming in. There's the phosphate attached. So this is beta-D-glucose 6-phosphate. Okay? This intermediate appears in glycolysis, the breakdown of glucose. Okay. You probably also have heard the term glycosides. Glycosides are sugars that have been altered at their anomeric carbon. A glycoside is something that's been altered at its anomeric carbon. You can see this anomeric carbon had an OH. This H has been replaced with a methyl group, and that creates what we call a glycosidic bond. Glycosides have glycosidic bonds, and no, I don't care if you be able to write methyl BD, glucopyranoside, a glycoside. Okay. The names can get kind of hairy. I try to keep it as simple as we can. Okay. But anytime I alter the hydroxyl on an anomeric carbon, I create a glycoside. Okay. Here's an important glycoside, laetrile. Laetrile is a poison that is found in apricot pits, among other things. And some people think it's a magic drug, which it really isn't. They think it's a magic drug because it actually is a poison. That's cyanide right there. And cyanide can be released, and probably not something that you want to have happen. It's also found in almonds. And no, it's not found in an abundant enough source in almonds to kill you. Okay. You guys are quiet today.
You said laetrile is a glycoside?
Laetrile is a glycoside. Yeah. I'm going to stop. Questions? I'll let you catch your breath. Give your arm a rest. Yes, sir? Why is cyanide poison? Very good question. Cyanide stops the process of electron transport. And electron transport is how we make 99% of our ATP. So by stopping electron transport, we stop ultimately the process of oxidative phosphorylation where the ATP is actually made. So cyanide is very potent at that, unfortunately. You guys want a joke? Okay. All right, so this is my dean joke, all right? I like to tell jokes about deans, all right? Hello, deans. Okay. You know they watch my lectures, right? If they do, I'm probably dead. Okay. All right, so there's these 3 deans. Or I guess it just takes 2 deans for this joke. But there's these 2 deans, all right? And they're sitting on this airplane. Have you heard this joke? Oh, man. You want to tell it? No? Don't give away the punchline, okay? Okay, all right. So there's these 2 deans, and they're sitting on this airplane, all right? And they get up, and the pilot says, "This is your pilot speaking." He says, "We are flying from Los Angeles to New York, and we plan to arrive at New York at midnight tonight." And the one dean says, the other says, "Oh, great, we'll get a little rest before we get there and so forth, and that'll be fine." And they're flying along for a little while, and all of a sudden they hear this bang! And one dean says there, "What was that?" "Well, I don't know." Pilot goes, "Oh," he says, "well," he said, "you probably heard that little noise there." He says, "Nothing much to worry about." He says, "Our number 4 engine just kicked out, but he says, we still have 3 engines, and we will be arriving in New York at 1 a.m. He says, "Well, we're going to get there at least. It's not a big deal, right? So we still have 3 engines." And a little bit later, they're starting to nod off, and all of a sudden they hear this bang again. The pilot says, "Whoa," he says, "I can't believe this is happening, but we have number 3 engine that just kicked out, and we still have 2 engines, and we'll arrive in New York at 2 a.m. Dean says, "Well, okay," and they go back to sleep, And a little bit later, bang! The pilot says, "This is really weird. So I've been on this, done this flight many times, never had, this happen before, but number 2 engine has just conked out, and we still have 1 engine left. I hope to arrive in New York by 3 a.m. And one dean looks at the other dean and he says, "If that last engine goes out," he said, we're going to be flying all night." I should have told it in my high voice. That's what seems to usually work, but that didn't. I didn't think about that beforehand. Deans don't have high voices like that, I guess, but all right. Pilots don't either. "Hello, I'll be your pilot." All right. All right. Let's talk about disaccharides. I know you guys want to talk about disaccharides, right? Yes. All right. I hear you. We're going to talk about disaccharides. What are disaccharides? Disaccharides are composed of 2 saccharides. That is, they take 2 sugars and stick them together, and you have a disaccharide. That's how you make a disaccharide. Amazing, huh? Okay. So these 2 disaccharides have been joined together, and they've been joined together by what's called a glycosidic bond. Because, why was it a glycosidic bond? Because there was the anomeric carbon right there. How did I know that was the anomeric carbon? Because it was the one attached to the oxygen. And it got attached here. The hydroxyl got disrupted. It made a glycoside, and glycosides have a glycosidic bond. Is this guy a reducing sugar or not? Okay, I'll tell you, since I haven't given you probably enough information to answer that. The answer is yes. Okay? And the reason is because this is an aldose. That is carbon number 1. We don't see a carbon sticking up here. So an aldose could form an aldehyde, since it has its free hydroxyl group, that could oxidize and become a reducing sugar. So that is a reducing sugar, yes. Okay? This has what's called an alpha-1,4 glycosidic bond. Alpha meaning this O of the OH went down, and it linked to carbon number 4's OH over here and made that bond. Here is an alpha-1,6. It went here and then linked to the carbon number 6 up there. Okay? All right. Now, disaccharides are important. Okay? Here is that one of those hand-drawn figures that I make that is better than I get from any book. Okay? And it shows sucrose. Sucrose is a disaccharide of glucose and fructose. Now, you can actually see fructose on the bottom configuration here. I'm going to tell you about that in a second. All right? Here's glucose on the top. The reason I draw this is virtually every textbook I've looked in draws sucrose in a really stupid way. They don't show it like this. They put them side by side, and to do that, they have to flip one of the configurations upside down. So students look at it and they're always confused. So it makes much more sense to draw the structure, even if it's amateur, but to draw the structure showing glucose over fructose as it actually is here. Okay, now glucose on top. Glucose is in the alpha configuration, right? There's the alpha D-glucose. Fructose is in the beta configuration, all right? Why is it in the beta? Well, let's look here, all right? Here's carbon number 1. Carbon number 2 is the anomeric carbon, and the hydroxyl went up, right? So when the hydroxyl goes up, that's beta. So the beta for the fructose, alpha for the glucose, and then there's the rest of the structure of fructose down here. Sucrose is interesting in that it's a diglycoside, meaning that both the anomeric carbon of the glucose and the anomeric carbon of the fructose are joined. They're both in glycosidic bonds. So it's a diglycoside. Okay? Now, this also has an important effect. All right? The important effect that it has is this guy is much less reactive. It's much stabler. The reason that they sweeten things with sucrose and not with glucose is because this guy sits around for a lot longer. This guy is not nearly as reactive as glucose is. Okay? If I simply use glucose, then I have problems. Okay. Here are some other disaccharides, and I'm not going to ask you to draw these. Oh, by the way, I should say, yes, I do think you should be able to draw sucrose, and the reason you should is because I've already asked you to know the structure of glucose and fructose. So if you know glucose and fructose, all you have to do is remember the alpha and the beta, and you can draw this structure. I'm not going to ask you to draw the other disaccharides. Okay? I will not ask you to draw the other disaccharides, but I think you should know what's in them. Okay? Lactose in particular. Lactose is milk sugar. It is a disaccharide comprised of galactose and glucose. It's the sugar found in milk. It's the sugar that was what was activating the lac operon in bacteria, if you recall. There are some other ones shown over here. I don't think they're nearly as important as lactose. Now, at this point, when I talk about sugars, one of the most common questions I get is, well, what about artificial sweeteners? What do I think about artificial sweeteners? To be honest with you, I'm not very fond of artificial sweeteners except in very small quantities. Okay? Having a Diet Coke, one Diet Coke every few days is probably okay. Drinking a liter of Diet Coke every day, probably not a good idea. Okay? There are different kinds of artificial sweeteners. NutraSweet is a relatively innocuous one, and it's the most common one that you find in soft drinks, for example. But it should not be drunk by anybody who's phenylketonuria. I think I talked about that on the first day of class, because it contains the amino acid phenylalanine and can literally poison a person who has phenylketonuria. Okay, so you don't want to be taking that if you have that. There are some suggestions that NutraSweet in high quantities may cause problems for people's eyes, for example, because it can create, some reactions of it can create formaldehyde, which can be very problematic for retinas, for example. And the other one that's getting popular is sucralose. And sucralose is one that I really take some issue with, although to be honest with you, I don't have any compelling evidence that it's bad for you. Okay? Nonetheless, I'll tell you a story about sucralose that will tell you why I have a little bit of concern about it. Okay? So the story about sucralose goes as follows. Okay? People were interested in finding a way to kill insects. They were looking for an insecticide. They were trying to make an insecticide. And when they tried to make an insecticide, they decided that the way to make an insecticide was to do something, use a principle of chemistry that we know is poisonous. Dioxin, for example, is a very poisonous compound. And there are other compounds that have carbon-chlorine bonds. We don't see carbon-chlorine bonds appearing anywhere in nature, at least anywhere in nature that I'm aware of. They're a very unnatural bond, and they're not a bond that forms readily. You have to go and be an organic chemist to make a carbon-chlorine bond. And so these guys were deciding to make an insecticide, and so they went to the logical thing. They said, we'll make some carbon-chlorine bonds and put it into our insecticide to make it. And so they're making this thing. These guys are in England. They're making this compound, and I guess they worked for some time to make this compound. And this I don't know how much of this is urban legend or not, but I have it on reasonably reliable source. Okay? That is, they're making this compound. Okay? The professor in the lab finishes the synthesis and leaves a message on the phone for the graduate student who wasn't there when the synthesis was done. The professor goes home and leaves a message to the graduate student saying, I finished the synthesis of this. I want you to test it. And the graduate student misunderstood it and thought he said, I want you to taste it. And being a good graduate student, if the professor says taste it, what do? He goes and he tastes what was supposedly this insecticide. And when the professor sees him on Monday morning, he says, well, how did it work out? And he says, it tasted great. And he said, what do you mean? You tasted it? Yes, it was very sweet. Needless to say, that's how they got around to deciding that, first of all, it wasn't poisonous, and that, second of all, that it might be an artificial sweetener. Now, why do I think this might be problematic? I think it could be problematic, A, if it got incorporated in some way into your glycogen. Glycogen we'll talk about later. Second, the reason they were making this was because it was basically undigestible. Okay? Undigestible things in your body have a tendency to accumulate. Okay? NutraSweet doesn't have that problem. NutraSweet is a peptide. NutraSweet is readily broken down by enzymes in your gut. This guy, I don't know what happens to it. Okay? So I'm not so fond of sucralose, although again, I have no evidence that there's anything wrong with it. You may be perfectly happy and functional with it. Okay. Another thing about artificial sweeteners that I should point out, and this has been something that's been realized in just recent years, and that is that your body, when you take an artificial sweetener, when you eat an artificial sweetener, your brain gets fooled. All right? Your brain is getting fooled into thinking you're drinking something sweet, but your brain plays a role in telling your body that we're getting ready for a meal. So what your brain ultimately does, we think, and there's evidence that it's happening, is it starts stimulating the production of insulin to get ready to handle that load of glucose that'll be coming with the meal that it thinks you're eating. All right? So your brain is anticipating food coming, food's not coming, your insulin levels go up. What happens when your insulin levels go up? Well, when your insulin levels go up, your body starts taking glucose out of the bloodstream. And when glucose starts going out of the bloodstream, what happens? Your blood glucose levels fall, and you get hungry. So drinking artificially sweetened things may, in fact, not work quite as well as you think they do. In fact, there's some evidence that drinking artificially sweetened things causes people to eat more to make up for it because, again, they feel this loss of glucose that's happening from the fact their body is producing insulin. Anyway, that's anecdotal, but that's an interesting perspective on that. Okay. Today's the day for stories. Polysaccharides. Polysaccharides, as the name suggests, contain many sugar residues joined to each other, sugar units joined to each other. And amylose is one in which all of the units are joined together in an identical fashion. All the units are glucoses. And the glucoses are all joined in an alpha 1,4 configuration. I think that's worth knowing. All the units are joined in an alpha 1,4. So it's a polymer of glucose, all with identical structures. Okay? Glycogen is a related compound. Amylose is found in plants, by the way. Okay? Glycogen is a related compound that is a storage form of glucose for animals. So amylose is a storage form for plants. Glycogen is a storage form for animals. Okay? Now, glycogen is not identical to amylose. It is a polymer of glucose, yes. And yes, it has many sugars joined with alpha-1,4 linkages, but it also has some branches that are alpha-1,6. There's a 6, there's an alpha-1,6 bond right there. Okay? We can think of amylose as being one long linear chain. We can think of glycogen as being a linear chain with all these branches coming off of it, very much like a tree has branches that go extensively. Okay? Now, we'll see later why those branches are very important for animals and why plants don't have very many of them. Plants have a compound related to glycogen called amylopectin. Amylopectin, A-M-Y-L-O-P-E-C-T-I-N. Amylopectin is like glycogen. That is, it has a polymer of glucose, it has alpha-1,4 bonds, and it has alpha-1,6 branches, but it doesn't have nearly as many branches as glucose does. I'm sorry, as glycogen does. It doesn't have nearly as many branches as glycogen does. Glycogen has branches about 1 in every 10 glucoses. Amylopectin has 1 in about every 30 to 50. So it's not nearly as branched. So I've just given you 3 polysaccharides. Amylose, which has no branches, storage form in plants. Amylopectin, which is also a storage form in plants that has some branches of 1,6. And glycogen, which is a storage form in animals, which has many branches, 1,6. If you take amylose and amylopectin and you mix it together, you create something that you all refer to as starch. So starch isn't a single thing, it's a mixture of amylose and amylopectin. Okay. I'm going through this fairly quickly. Another polysaccharide, okay, is related to amylose. And it's also found in plants. And it's cellulose. Cellulose is also a polymer of glucose. All these guys are polymers of glucose. But instead of being alpha-1,4 linkages, cellulose has beta-1,4 linkages. You see that oxygen right there is going up, not down. That is a beta linkage. Cellulose is a polymer of glucose with beta-1,4 linkages. Okay, now, that very trivial thing is very, very important because we have enzymes to break down amylose. We have enzymes to break down amylopectin. We have enzymes to break down glycogen. We have no enzymes to break down cellulose. The reason roughage is because it has cellulose that our digestive system can't digest, and it passes through the digestive system fairly readily. Okay? That's what roughage is. Okay? Ruminant animals, on the other hand, like cows, have a bacterium in their rumen. The rumen is a specialized stomach that cattle have. They have a bacterium in them that contains the enzyme cellulase. Cellulase. Okay? And as its name suggests, cellulase breaks down cellulose. Okay? Because of that, plants can eat grass, digest the cellulose, and cellulose is a component of the cell walls of plants. That's why we see it so abundantly in plants. Okay? They can eat grass and get a lot of energy out of the grass because all of those cellulose molecules that are in the grass can be broken down into glucose, and the cow can use that for energy. We can't do that. Okay, starch, mixtures of those guys, amylose and amylopectin. And amine sugars. Okay. As its name suggests, there's a sugar. They've got an amine group attached. Most commonly, the amine group is attached at carbon number 2. And there's a variety of different structures that can arise as a result of that. Okay, I feel like I missed something up here, but I don't see it. What's that? Oh, lactose intolerance. Okay, I pointed to it and I didn't tell you about it. Lactose intolerance, yeah. So lactose intolerance arises in people because as we get older, we no longer make the enzyme lactase. Okay? That varies from person to person, and it varies typically among ethnicities. Asians tend to have more lactose intolerance than non-Asians. All right? And if you think about it, the most natural thing in nature is to be lactose intolerant, right? Because lactase is there for infants that are nursing. Only in human beings do they drink milk and dairy products as they get older, okay? Well, during the time humans were evolving, they weren't doing that for the most part, okay? And so adults, I mean, it's sort of built into us, the older we get, the less lactase we're going to make. So lactose intolerance arises because we don't digest lactase into galactose and glucose, and consequently it travels further in our digestive system to a place where bacteria go crazy, where they normally don't get that chance. So if you have lactase present, you break it down before it gets very far into your intestines. Okay? If you don't make lactase, then what happens is it travels further into your intestines, and some bacteria get a hold of it and go, whoopee, there's the whoopee again, because they start breaking it down and making a lot of gas. And it's very uncomfortable and can cause real problems. There are things that you can take. There are capsules that you can take of lactase that will help you with lactose intolerance if you are lactose intolerant. And that's one thing there. Okay. I have a song to explain things. So let me go through that, and then we'll call it a day. The song is very easy to sing. It's very short, too. It's called The Heart to Sucrose. Carbohydrates all should sing, glory to the Hayworth ring. Anomeric carbons hide when they're in a glycoside. Glucopyranose is there in the boat or in the chair. Alpha, beta, D, and L, diastereomer hell. Alpha, beta, D, and L, diastereomer hell. Okay, have fun, guys.